Hello, my name is Lauren Brandt, and my project tested the effect of heavy metal exposure on limitations of insulin-producing beta cells and the trigger of diabetes mellitus. To start off, there are about 1 in 500 people in the US that have the condition, and about 80 people get diagnosed with type 1 diabetes each day. Insulin therapy is currently the only effective treatment for the disease, and the disease costs $14.9 billion in healthcare costs per year. 5% of people who have a parent or sibling with type 1 diabetes will also develop the condition. This shows how important diabetes research is, as so many people are dealing with the disease and how costly it truly is. So you may be asking, what causes type 1 diabetes? Type 1 diabetes is caused by the immune cells in your pancreas destroying the beta cells that are in the pancreas. So this causes your pancreas no longer producing insulin, which causes glucose in the blood to increase as it can't be absorbed. This causes high blood sugar and many other problems. I decided to do my research on environmental factors, and some previous research was done on 2 heavy metals, the first one being arsenic, and it was shown that arsenic actually had disturbed the movement of glucose in the body, as well as disturbed B cell function, which causes the onset of type 1 diabetes. The other heavy metal was mercury, and mercury showed a high likelihood to affect B cell survival and function. So both of these are affecting the B cells in the pancreas, which cause insulin to no longer be secreted, causing type 1 diabetes to be developed. I chose the heavy metal nickel for my project as there wasn't a lot of research done on it, and nickel is frequently found in smaller concentrations in everyday items like food and water. This is actually a continuation project. So last year I asked, will 1 micromole nickel cause a decrease in insulin secretion by pancreatic beta cells? It actually did. So the data supported my hypothesis, and this is my graph. So it showed that the control had a much higher, whereas 1 micromole had a lower insulin secretion, showing that as the cells were exposed to nickel, then it caused less insulin to be secreted. This caused me to ask the question about why is this happening, so that influenced my research this year, as well as I wanted to test further concentrations of nickel. So why is this important? It's important to understand the causes of why this is happening and the causes of type 1 diabetes in general. It helps doctors identify triggers for the disease and prevent development of the disease in the future. So my hypothesis was, if insulin-producing beta cells stop functioning, then type 1 diabetes will develop. So if functioning beta cells are exposed to 135 micromole of nickel, then lower insulin secretion will happen as nickel concentration increases, as well as lower cell proliferation as nickel concentration increases, and finally lower cell viability as nickel concentration increases. And all of these factors are showing that signs of type 1 diabetes is developed. So in order to do my project, I had 12 cultures of the beta cells, so 3 for control, 3 for 1 micromole, 3 for 3 micromole, and 3 for 5 micromole. After they were exposed to the media that they were, which was either the control, the 1, 3, or 5 micromole of nickel, I exposed them for 96 hours, and then after the 96 hours, I took the cells or the media and exposed them for an ELISA assay to measure insulin secretion, or an MTT assay to measure cell proliferation, or I used trypan blue and a hemocytometer to measure cell viability. So first was insulin ELISA. And to do this, I took the media from the cells and I put them in a 96 well plate of this ELISA assay, which came with an insulin antibody already attached at each well plate. So this caused the insulin in the media, the insulin antigen, to attach to the insulin antibody. So after it was washed away and more chemicals were added, then the insulin antigen would stay there. This allowed for, once the plate was put into the microplate reader, it was able to read the insulin absorbances. For the MTT assay, it's a similar process because it's a 96 well plate assay, so the cells were plated, so I had to make sure there was about the same amount of cells in each plate. Then the MTT reagent was added, and then the absorbance were read at 570 nanometers. So as you can see with the picture, with MTT, it's a purple precipitate, so a darker precipitate, then there's higher proliferation. Finally, I did trypan blue to measure cell viability. So this was dyeing the cells and counting the dead to live ratio. My results are here shown. With my ELISA assay, the insulin secretion, it did show that as nickel concentration increased, then there was less insulin secreted. MTT assay, there were no trends shown, as well as cell viability, there were no trends initially shown. So going to my discussion, I figured I needed to explain the control in more detail, as the control was actually the opposite of what I expected. It was always the lowest. It was the lowest value in the ELISA, MDT, and cell viability test. This was the opposite of what I was expecting. I expected the control to be the highest as it was not exposed to nickel, because I was thinking that the nickel would cause everything to be lower. This could have actually occurred due to a lab error. When I was looking at the data more in depth, I saw that the 3 micromole sample kind of looked like what the control should look like in some situations. This makes me wonder if I accidentally switched them during some testing. And then the control cells may have been less healthy compared to other cells. This could have been done to growing the cells to about 100% confluency, and as well as during the subculturing method, the control were what I ended up starting with. So they could have been contaminated or just not as healthy because they were older cells. Moving on to insulin secretion. So to just remind you, I used an ELISA assay to measure the amount of insulin secreted by each sample. As nickel concentration increased, insulin secretion did decrease, and I did an ANOVA test, and it showed that the results were significant. I also did a t-test between the 1 micromole and 3 micromole sample and the 1 micromole and 5 micromole sample, and this was also significant, showing that there was a difference between the two. And this ultimately shows that nickel concentration does affect insulin secretion. And moving on to cell proliferation in the MTT was conducted to measure the cell growth of the samples, and the results were not expected, showing no trend in cell proliferation. My ANOVA test showed that there was no significance in the difference. This data just means that nickel does not impact cell proliferation and can be ruled out as a cause for decrease in insulin secretion. And finally, cell viability. The cell viability was tested to measure how many cells were still alive compared to dead after nickel exposure. And again, there was no trend in cell viability. And I did a chi-squared and an ANOVA test were both conducted, and they both were not found to be significant. And these results show that cell death is also not a factor into why there's less insulin secreted by the cells. So it makes you wonder what is causing the insulin secretion. For my conclusion, insulin secretion, the initial hypothesis was supported where insulin secretion decreasing with increased nickel concentration. However, the control did show the lowest amount of insulin secreted where it was hypothesized to be the highest. Although, like I stated earlier, this could be due to the age of the cells or another lab error. In cell proliferation, the hypothesis was not supported, where the cell proliferation decreased from 1 micromole to 5 micromole. However, it increased to 3 micromoles, so there was no overall trend. This concludes that nickel concentration does not impact cell proliferation. And cell viability, the hypothesis was also not supported. There's no trend in cell viability, and this concludes that nickel concentration does not impact cell viability. In the future, it's important to test other factors. So I saw that it might be important to test a disturbance in the cell signaling pathway. And to define cell signaling is the communication between cells to release hormones and other signals to regulate the body. In one of the articles that I had researched and background research, it was found in arsenic exposure that there was an activated signaling pathway which caused the decrease in insulin secretion. And pretty much what was happening was the arsenic was telling the cells there was enough insulin in the body when there actually was not. This caused blood sugar to rise as not enough insulin was being secreted, causing type 1 diabetes. Even though the results of my project were not as expected, it can continue to help research towards the cure of the disease and allow people to be more cautious knowing possible causes of the disease. And here are my resources. Thank you.